So, dear students, so today our topic is enzymes action. So, before going to the enzyme action, we should be learn regarding the enzymes. So, already you know the what are enzymes, so their application, types of enzymes, so many more regarding the enzymes. Because enzymes, so they are the biological catalyst. Okay, it means they can catalyze the various reactions in the biomolecules. So they convert larger biomolecule into smaller biomolecule, as well as they can utilize the smaller biomolecule then involved in the synthesis of the larger biomolecules. So the enzymes, so they are mainly known as the catalyst, and they involved in the conversions or modifications of the biochemicals. So that's why, so they are known as the biological catalyst. So enzymes, so usually, so they are made up of the proteins. Okay, sometimes, so enzymes are RNA nature or RNA, both RNA or protein complexes. So enzymes, so those are present in the digestive tract. Same enzymes is also found in the blood. In the blood, so they are present in the inactive forms, but they are only active, so in the digestive tract, so were they involved in the digestions of the food, so regarding the carbohydrates, fats, as well as the proteins. So in the digestive tract only, so they are active, and same enzymes is also found in our blood streams. So but were, so they are present in the inactive form, and such enzymes are known as the zymogens or proenzymes. So those enzymes in GI tract, so they will be active, but in blood, so they are inactive, and such enzymes are known as the proenzymes or zymogens. So regarding the active sites, so as you know, so the enzymes are made up of proteins, and proteins, so they are made up of the amino acid. So long chains or amino acid, they are joined together. So with the help of the peptide bond, so there forms the chain. So those chains are known as the peptide. So those are responsible for formations of the enzymes. So enzymes are nothing but, so these are the group of amino acids, or they are made up of the polypeptides. And here the polypeptide, so they are formed from the series of amino acids with the help of the DNA and with the help of the RNA. So they are involved in the synthesis of the enzymes. And these amino acids, so those are present in the enzymes, so they act as a active sites. Okay, active sites on the enzymes are there nothing but, so whatever the components, so they are present in the enzymes like amino acids, so those amino acids, so they are acts as the active sites. And here the active site, it is an place or surfaces on the enzyme. It is helpful for, or it can allow the binding of the substrate. Okay, so enzymes, so they are made up of amino acids, and these amino acids, so they acts as the active sites, and active sites is the place or surfaces it can allow the to hold or to bind the substrate. So after enzyme or substrate can bind with the active sites, then only the enzyme substrate complex will be forms, then only the enzymes can catalyze a biochemical reactions. So that's why, so active sites on the enzyme is required for the conversions of substrate, that is a larger particle into smaller particle, or one product to the another product. So that product is required for the body, that only possible, so when the substrate will be bind on the active site, those active sites, so those are present on the enzyme surfaces. And another terminology regarding the halo enzyme, some enzymes are known as the halo enzymes. Halo enzymes means, so they require the non-protein part to show the certain actions or to regulate the biochemical reactions where they are required the non-protein parts, may be coenzyme, may be cofactors, and such enzymes are known as the holoenzymes. Holoenzymes are nothing but, so these are the composition of apoenzymes plus cofactors. Here cofactors may be minerals or metals like zinc, magnesium, calcium, many others are there. So they act as the cofactors, or certain coenzymes are there. So in the presence of those coenzymes and metal cofactor, then only the enzymes are involved in the catabolic reactions or anabolic reaction, and such enzymes are known as the holoenzymes. So here the holoenzymes, this is the combination of apoenzyme plus cofactor, so that can yield, that is the haloenzyme. Haloenzyme nothing but, so they are only active in the presence of the certain non-protein molecule or non-protein substances, like maybe cofactor, maybe coenzyme. So here apoenzymes, so apoenzyme is an inactive enzyme. It only active when it is having the organic inorganic cofactors. Okay, so apoenzymes, so these are the one type of enzymes. So they are always present in inactive. So when they are active upon the binding to the organic or inorganic cofactors, maybe calcium, maybe sodium, manganese, especially that is zinc. So in the presence of zinc, so when the enzyme bind to the organic inorganic cofactors, so then only, so that enzymes becomes the active, and such enzymes are known as the apoenzymes. Okay, and proenzymes are zymogen, so these are present and inactive in the blood, so those are known as the apoenzymes. Now, okay, proenzymes and apoenzymes. So these are active only when they are binding to the organic and inorganic cofactor haloenzymes. So these are required both apoenzyme as well as the cofactor. So then only, so they are active, they can regulate the catabolic reactions. And here the active sites. So these are nothing but, so they are special surfaces made up of the group of proteins. So they allow to bind the substrate on the enzymes. So they are responsible for the formations of the enzyme substrate complex. So this complex is needed for to catalyze the enzyme various biochemical reactions, and those are surfaces are known as the active sites. So next one is the, so regarding mechanism of enzyme actions, so already you know, so enzymes having the active sites, so where the substrate can be bound. Okay, then after binding of enzyme substrate complex, so there is the catabolism of weak bond will be taken place, especially the hydrogen bond that can be catabolized, or many other bonds are there, so they can break down. Okay, due to formations of the enzyme substrate complex, and this enzyme substrate complex is mainly depending upon the concentrations of enzymes and as well as active site, so those are present on the enzymes, as well as the concentrations of the substrate. Even they may be depending upon the cofactor, ability of the cofactor as well as the coenzyme. So here the substrate, it can bind on the active site, so that site was formed due to presence of amino acid chain on the enzymes. It can allow the binding of substrate, then it can form the enzyme substrate complex. Then after that, the enzyme, it can break down the various weak bonds, hydrogen bonds or many other bonds or peptide bonds, many bonds are there. So they're weak in nature, so they can decrease the energy level, then it can forms the product. So that is the where the enzyme can separate it from the product. So like that, so the enzyme actions can be occurs after only the substrate can be bind to the active sites on the enzymes. Okay, so this mechanism of enzyme action was mainly demonstrated by two scientists. One is the Fisher, so that model is known as the lock and key, and another one is a Coastland. So that model was known as the induced fit models. Okay, so regarding the lock and key, so here the substrate, it can bind on the active sites, it can forms the lock and key. So then the lock is closed, so that was for responsible for the formations of the enzyme substrate complex. So after that only the catabolic reactions or anabolic reactions are taken place, then the product 1 and product 2 are forms by subject to the catabolic actions of the enzyme. So that was demonstrated by the Fischer. That model was known as the lock and key model. And another scientist, that is the Costland, he also explains regarding the mechanism of enzyme action. So that was known as the induced fit. Okay, here the substrate, so they can bind to the active sites that is present on the enzyme in the both, that is one site and two site. Okay, it's substrate, there is another chain, it substrate chain that will bind to the another site. Then various catabolic reactions occurs, so that can forms the enzyme substrate complex. Then catabolic reactions occurs, then weak bond can be breakdown, especially that is hydrogen bond and later on the peptide bond. So then that was responsible for the catabolic action, is responsible for the formations of the product 1 and product. So like that the enzymes, it was involved in the conversions of larger molecule into smaller molecule, or it can neutralize the smaller molecules and involved in the formations of the larger molecules. So this is regarding the mechanism of enzyme action. So next one is the, that is the flow charts regarding the catabolic cycles of an enzyme. So first enzyme, it can penetrate into the enzyme, the substrate, easily it can penetrate inside the enzyme. Then enzyme, it can structurally modify, it can modified as per need of the substrate to bind on the enzyme. So that was responsible for the formations of the enzyme substrate complex. After that, these interactions is occurs between the enzyme and substrate, such as on the site of hydrogen bond and as well as the ionic bond. So after that, various structural changes can be taken place. So where enzyme break down the larger particle into a smaller particle, then finally it can convert substrate into the products. Then the products, so they are released from the enzyme. Then enzyme is once again, it can free for attachment of the another substrate. So this is the catalytic cycles regarding the enzyme actions, where substrate can bind on the active site. Later on, various interaction is occurs. So then structural modification occurs on the substrate, then substrate can convert into product, then the product are released, then they are removed from the reactions. Then so that enzyme was free for to hold the new substrate molecule for the catabolic reactions. So next one is the regarding the progress. So here this is one example regarding the catabolism of H2O2 into a oxygen and hydrogen. Okay, so this H2O2 can be catabolized or break down into products like oxygen, water. So in the presence of enzyme, the shorter duration is required. Okay, and as well as the energy is required for this catabolic reaction is the less as compared to without enzymes. Okay, so without enzymes, then also the H2O2 can convert into oxygen and water, but it required the more energy as compared to the in the presence of enzyme. So that's why, so enzyme, so they can accelerate the rate of reaction, as well as, so whatever the expenditures or energy was required, that also required the less, even the time will be required less, the formations of the product. So that is the applications of the enzymes or the rule of the enzymes in the various catabolic reactions in the presence of enzyme. So that reactions can easily take place without that much of lot of energy, and as well as the time required for reaction, so that was also decreases. But without enzyme, the activation energy is required more, as well as the time is also required more. So that is the one of the main applications of the enzymes in the various catabolic reactions. So next one is the enzyme inhibitions. So here certain substances are there. So next topic is a enzyme inhibition. So here certain substrate or substance are there, so they are inhibit the catabolic activity of enzyme. So that phenomena was known as the enzyme inhibition. So water substance, so those are involved in the inhibitions of the enzyme activity, so those are known as the enzyme inhibitors. So regarding definition, so enzyme inhibitor is defined as the substance which binds with the enzyme and brings about to decrease the catalytic activity of that enzymes. So here, so these inhibitors, so their structure similar to the substrate. Okay, certain substances are there, so their structure is similar to the substrate. So due to that, instead of substrate, so these substances, so they are bind to the active sites. So due to binding of these substances or enzyme inhibitor, the catabolic activity of enzyme is decreases. So that was known as the enzyme inhibition. So whatever substances, so they are required, so they are involved in the to decrease the catabolic activity of the enzyme. So those are known as the enzyme inhibitors. So enzyme inhibitors, so they are nothing but, so these are also substance. Either the structure is similar to the substrate, or so they are modify the active sites, or so they are diminishes the whatever the cofactor and coenzymes are required, so they are decrease the level. So without cofactor, without coenzyme, so that enzymes activity can be decreases or cannot be products or form, and such substances are known as the enzyme inhibitors. Okay, so any substance that can diminish the velocity of enzyme catabolic reaction, those are also known as the enzyme inhibitor. So these enzyme inhibitor may be drugs, may be antibiotics, may be poisons, may be anti-metabolites. Okay, certain enzyme inhibitors are the drugs. Okay, for example, nimesulide, okay, or paracetamol. So this paracetamol, it mainly helpful for the treatment of inflammations, and inflammation is the one type of the biochemical reaction that can occur in the body, so where the various autocoids are formed. So those autocoids, so they're involved in the formations of swelling, as well as wound, and as well as irritation, itching. Okay, and as well as, so they're involved in the form of the inflammation by one enzyme. So that enzyme was known as the cyclooxygenase enzyme. So here the drugs like paracetamol and nimesulide, it target that enzyme, it can inhibit that enzyme. So due to lack of that enzyme, the inflammation process or accumulation of fluid at the site of injury or inflammation, so that was do not occur in the presence of drug paracetamol, or absence of the cyclooxygenase activity by inhibitions of the drug like a paracetamol, nimesulide, any other drugs are there. Okay, so due to that, that was helpful for to treat the inflammations. So the antibiotics are there, so they mainly kills the microorganisms by inhibiting the active metabolite enzymes in the bacterial wall. Okay, so in the bacterial wall, so various enzymes are involved. So those enzymes are required further to maintain the life of the bacteria, or to repair the damaged plasma membranes of bacteria. Without those enzymes, so those microorganisms cannot be survived. For example, penicillinase is an enzyme. So it was present in the various gram-negative bacteria and gram-positive bacteria. Okay, so those bacteria, especially E. coli, it is involved in the cause of the fever, food poisoning, and inflammations, and many other disorders. Okay, so that E. coli life was mainly depending upon the penicillinase enzyme. So here the antibiotic like penicillins or many others are there, amoxicillin, then coloxycillin. So they are inhibit the, so those enzymes, then they can helpful for to ride out the E. coli induced inflammations or food poisonings. Then as well as poisons. So poisons mainly they are inhibit the ATP source by targeting the various enzymes like glucokinase or many enzymes are there. So due to entry of poisons, so they are inhibit the enzymes. So those are involved in the productions of the ATP, as well as so the anti-metabolites are there. Their structure is similar to the enzymes components like a purine and pyrimidine bases. Due to that, so they inhibit the metabolic activity in that particular type of cell. So that's why the anti-metabolites, so they are mainly employed for the treatment of the cancers. Okay, the cancer, so where the abnormal growth of cell is present. Cell rapidly growth as compared to healthy cells. So then there is responsible to formations of the tumor. And here the antimetabolite, the structure are these are nothing but these are type of chemicals or drugs. They structure similar to the purine and pyrimidine bases like thymine, guanine, cytosine, uracil. So due to that, they inhibit the formations of proteins in the cancer cell. So due to that, they are helpful for treatment of the various types of the cancer. So that's why the enzyme inhibitors are drugs or substances, antibiotics, poisons or anti-metabolites. So they also decrease the beneficial effect as well as they helpful for to excel the beneficial effect. If it is biochemical reaction means where it can be decreases. But in case of certain diseases where it can show the beneficial effect by inhibiting the enzyme active and such substances are known as the enzyme inhibitors. Okay, so here the enzyme inhibitors may be organic or inorganic in natures. They having the both nature, there is the organic as well as the inorganic in the natures. So and here, so these enzyme inhibitors, so they are mainly classified into three. That is the reversible inhibitions, irreversible inhibition, then allosteric inhibition. Okay, allosteric, so they are alter the allosteric site, those are present on the enzyme. So due to alterations of the allosteric point or site on the enzyme, they can inhibit the various enzymic reactions. Okay, the regarding reversible, those substance in the presence of those substance, the product cannot be formed. Once the substance is removed, then the product is continue the formations. Okay, or the enzyme that enzyme can be active, then it is involved in the formations of the product, and such reactions or such inhibitors are known as the reversible inhibitors, or that reaction was known as the reversible inhibitions. Then another is the irreversible. Once the substrate inhibit the enzyme activity permanently, so that enzyme activity can be blocked, and such substrate are known as the irreversible inhibitors, or that reactions was known as the irreversible inhibitions. So now we learn one by one. So first we learn regarding the reversible inhibitions or reversible inhibitors. So inhibitors, so they bond the covalently with the enzyme or in the same site, and they inhibit the binding of substrate to that active site. Okay, then so they can block the catabolic activity of the enzymes. So here, so these reversible inhibitions reaction, they are mainly classified into two types, that is the competitive inhibitions and non-competitive inhibitions. So competitive inhibitors, so they are bind on the active sites, and non-competitive inhibitors, so here the inhibitor, so they bind on the other than the active sites. But both cases, so they are inhibit the enzyme activity, and in the both cases, the product is not formed. Once you remove those substances, so those are involved in inhibition, then that reaction was or that enzyme activity was the preserved or continued, and such mechanisms was known as the reversible inhibition. So where in the presence of substrate or substance, they do not allow the binding of substrate to the active sites, then they decrease the catabolic activity of enzyme. Once that substance was removed means, then the as usually the enzyme is involved in the conversion of substrate into product, and such phenomena was known as the reversible inhibitions. And reversible inhibitions, once again they subclassified in two types, that is competitive inhibitions and non-competitive inhibitions. In a competitive inhibition, the substrate or substance are inhibitors, so they bind on the active sites, and in the non-competitive they do not bind on the active site, they can bind on the other site. So first one, that is the competitive inhibition. So here the inhibitor, the structure is similar to the substrate. So due to that, so they are bind on the active sites, then active sites is occupied by the substance or inhibitor. So that's why the substrate, they are failed to bind on the active sites. So lack of binding of substrate on active sites, the product do not forms. Okay, so those are known as the competitive inhibitors. So these competitive inhibitor, their structure is similar to the substrate. So instead of substrate, the enzyme it can allows the binding of enzyme inhibitor. So that's why, so where the enzyme and inhibitor reaction is taken plus, then finally, so there is no products can be formed. Once you remove the inhibitors, then the reaction was continued, and such phenomena was known as the competitive inhibitors, so where the inhibitors can bind on the same active sites, so where the substrates are bind due to structural similarity and shape. For example, the enzyme succinate dehydrogenase, also known as the SDH, is the example. Can inhibit the competitive inhibition of the succinic acid as it is substrates. Okay, so this compound, the namely the malonic acid, glutaric acid, and oxalic acid, they structure similar to the succinic acid. Instead of succinic acid, so these malonic acid, glutaric acid, oxalic acid inhibitor, they bind on the active site and they are inhibit the formations of the succinic acids or inhibit the activity of the succinate dehydrogenase. So such examples are known as the competitive inhibitors, and phenomena was known as the competitive inhibition. So where the inhibitors like malonic acid, glutaric acid, oxalic acid, they structure similar to the end product, that is the succinic acid. So succinic acid failed to bind on the active sites, so then the reactions can be retarded. So that was known as the competitive inhibitors. So next one is the non-competitive inhibitors. So here the inhibitor structure is not similar to the substrate. Even its shape is not similar to the substrate. Even the substrate can able to bind on the active sites. But non-competitive inhibitor, they do not bind on the active sites, so they are bind on the another site of an enzyme, then they inhibit the activity of enzyme, then the substrate cannot be converted into product, and such phenomena was known as the non-competitive inhibitors, of where some inhibitors they do not having any relations or structure similarity toward a substrate, but they bind on the another site and they inhibit the activity of enzyme either by slow down the enzyme activity, either by decrease the cofactor attachment to the enzyme, or they inhibit the activations of the coenzymes as well as enzyme. Then the substrate cannot be involved in the formations of the products. So such phenomena was known as the non-competitive inhibitions. So there is no structural similarity between the substrate and the inhibitors. So next one is the irreversible inhibition. So once the inhibitors are inhibit the enzyme, it means permanently. So that enzymes become the inactive. So that was known as the irreversible inhibitions. For example, the toxic substance in the case of poisoning, so they are inhibited the various enzymes permanently. Okay, and such phenomena was known as the irreversible inhibitions. Example is a iodoacetate. It is an irreversible inhibitor of enzyme like papain and glyceraldehyde 3-phosphate dehydrogenase enzymes. So these are two enzymes. Glyceraldehyde 3-phosphate dehydrogenase involved in the glycolysis, and papain is involved in the digestions of the food. And the presence of iodoacetate in our food, it permanently it can inhibit the activity of papain and the activity of the glyceraldehyde 3-phosphate dehydrogenase enzyme. So due to that, the digestion is decreases or the productions of ATP was decreases, and such are known as irreversible inhibitors. Another example, that is the disulfiram. It is an drug. So this drug was used for the treatment of the chronic alcoholisms. Okay, so this enzyme, this drug is mainly inhibit the one enzyme that was known as the aldehyde dehydrogenase enzyme. So this enzyme is required for the metabolism of alcohol. So alcohol, it can be metabolized into aldehyde in the presence of alcohol dehydrogenase enzyme. Then aldehyde metabolized in the presence of aldehyde dehydrogenase enzyme that can convert it into water and as well as the carbon dioxide from the aldehyde or acetaldehyde, and acetaldehyde forms from the alcohol. So when you are consuming the alcohol, So that alcohol is metabolized in the presence of alcohol dehydrogenase, then form into acetaldehyde. Then acetaldehyde is metabolized into carbon dioxide and water in the presence of aldehyde dehydrogenase enzyme. So the disulfiram, it is used for to withdraw the alcohol habits in the chronic patients. If you want to withdraw the alcoholic conditions or alcoholic habits in the any patient's person means, so there is a choice of the drug is the disulfiram. So this drug inhibit the, so this enzyme irreversibly. Then after that, if the same person, if they're taking alcohol means, so that alcohol converted to acetaldehyde, but the acetaldehyde cannot be metabolized into carbon dioxide and water. So due to that, the presence of disulfiram, the acetaldehyde concentration is increases in the blood, and that increased aldehyde concentration in the blood, it can cause a certain fear or tightening of the chest, or it can increases the suddenly rate of respiration and decrease the rate of respiration. Suddenly it can increases the heart rate, decrease heart rate. One type of fear it can create in the patients. Okay, by the use of disulfiram, by inhibiting the metabolism of acetaldehyde into carbon dioxide and water. So due to that fear, so that patients can withdraw the habit of the alcohols. So that's why, so disulfiram is the irreversible inhibitors. So that was used in the treatment of the chronic alcoholism, where it can irreversibly, it can inhibit the activity of aldehyde dehydrogenase enzyme. So that enzyme is required for the metabolism of the alcohol. Next one is the allosteric inhibitors. So these are also molecule or substance. So they are bind on the allosteric site. So it is another site. So they are present in the on the present on the enzyme, and they can modify the activity of the enzyme, either decreases or either increases. So for example, so an enzyme involved in glycolysis like phosphofructokinase. So this enzyme convert ADP into ATP. So when the ATP concentration is increases, so this ATP is acts as a allosteric inhibitor. It binds to the phosphofructokinase and inhibit the conversions of ADP into it is ATP. So like that, so this is ATP here acts as the allosteric inhibitor. So it can forms by fructokinase. Okay, so now another allosteric inhibitor, some are known as the positive allosteric inhibitors or regulators. So they can help to enzyme to work better or promote or stabilize the active conformations those are present on the enzymes. Okay, so those regulators are known as the positive allosteric regulators. So positive allosteric regulators or allosteric inhibitors nothing but, so these are the certain components or molecule. So they binds on the allosteric site and modify the activity of the enzyme, and such phenomena was known as the allosteric inhibitions.